that are coming up. And our first one, which is coming from Clément Garin, is on primate resting state networks. So a comparison between human and primate resting state networks, which is going to be very, very interesting to see. So without further ado, Clément, the floor is yours.
Sure, thank you very much. I'm going to start to share my screen. So you should now see it.
Yes, we do.
Great.
And then if you can, yep, lovely. Thank you.
Thank you for the very kind introduction. Today, as you said, we're going to talk about non-Hominid primate network, and especially we're going to focus on the default network that we're going to try to compare this default network from the primate to the human one. So as you may know, the default network is a network that is particularly involved and is particularly involved in high cognitive abilities such as self-directed thought. This network is highly active at rest, and we can actually divide this network in 5 clusters in which we can find the parietal cluster, the temporal cluster, the posterior cingulate cortex cluster, and the medial prefrontal cluster. The medial prefrontal cluster is actually an important cluster since it has been shown to be involved in a gateway function of self-representation in humans. In mammals, some default mode network-like has been proposed. One of them has been explained, has been detailed by Garin et al. In 2019 on what we can see here is that a seed, so meaning a small region, has been created in the anterior cingulate area, and after we can analyze a correlation to this region. So that's what you can see, and this is a default mode network that has been proposed in that species. You can see that we have some parietal temporal region that are correlated to this seed, as well as some frontal region. Exactly the same thing has been done a long time ago in rats. And in macaques, since we have a much bigger brain, we are able to actually have a bigger scale and to see with much more detail the region involved in this network. So in 2011, some clusters such as the medial prefrontal cortex, PPC, PCC, and dorso-lateral prefrontal cortex has been demonstrated to be involved in the default mode network-like of the macaques. More recently, in marmosets, Liu et al. Found that the medial prefrontal cortex seemed actually to be missing from the default mode network-like of this primate. So what they have done here is that they analyze the various correlations that they can observe between few seeds. You can see that, for example, here, the correlation between the PCC and the dorsolateral prefrontal cortex seems to be high, especially when compared to the correlation between the PCC and the medial prefrontal cortex. So that is actually very interesting for the function that I described earlier. So this result has actually also been confirmed with tracer injection that you can see here on your screen. So what the purpose of my work here? I could create one big question, that is the medial prefrontal cortex role in the default mode networks further developed in human than in other primates? So to do so, the first thing that I have done is to try to compare a statistical tool, a statistical analysis that will allow me to compare the role of the medial prefrontal cortex through different species, because of course we have different signal, we have different SNR, so it's actually more difficult than we think. Then I'm going to try to answer to that question. In the third time, I think if the medial prefrontal cortex is not involved in the default mode network in non-Hominid primate, in which network is it involved? So I'm going to also try to answer to that question. So here we studied 3 different primate species: human, macaques, and mouse lemur. Mouse lemur is a very small endemic species from Madagascar. And as you can see, the macaques were anesthetized, but not humans. So the first method that I choose to extract the network into the macaque's brain and into the human brains, and you will see not in the mouse lemur brain. This first part, I choose to use atlases, and these atlases actually display some homologous region, which is a great thing for comparison. And as I said, since mouse lemur doesn't have this atlas, I'm not gonna talk about the mouse lemur in this first part. So we extracted some seeds in various anatomical area of macaques on human. And as you can see here is a correlation, the statistical map that you can observe with similar region. And we use first the area 8AD, and you can see that we observe a region that we choose to call here the frontal-parietal network that seems to actually be very close to the literature and to what has been called the default mode network-like. In human, using a seed in the area 8AD, we obtain a default mode network, a classical one. Using a seeds in the medial prefrontal cortex, I obtain a network that links the medial prefrontal cortex or the 9D, but we also tested with the area, for example, 32. We have exactly the same network. We have a correlation of this region with some temporal area. When in human, we obtain the classical default mode network. Using a seeds in the area, anterior area of the cingulate cortex for 24C, we obtain again the frontotemporal network. When in human, we obtain the default mode network. And to the end, I choose to show you the area 46T that led also to the frontotemporal network in macaques, but to the executive control network in human. So you can already see that we might have some important discrepancy between the organization of the network in macaque and human. So the third species has been done to actually include all our three species, and I choose to perform on our bold images a dictionary learning analysis. This analysis is highly similar to ICA, but seems to have better decomposition of the overlapping signal. So classically, this method is going to produce statistical map, and this statistical map can be concatenated into functional atlas, as has been done, for example, by Yeo et al. In 2011. So since this study is a gold standard and find that we could segmented the brain network into 7 network in human, I choose to use 7 network in all our three primates. So here you can see the results in our 3 species, and I only show you 2 statistical map for each species, and you can see classically that we are able to extract the default mode network, the frontoparietal control network in human, and in macaques that we recovered the 2 networks that I described to you earlier, the frontoparietal network and the frontotemporal network. Interestingly, in mouse lemur, we find a frontoparietal network that doesn't seem also to involve the medial prefrontal cortex, and we find another network that involves the medial prefrontal cortex on some temporal area. So then we concatenated all these 7 statistical maps to build 1 functional atlas for each species, and we segmented all this cluster based on the anatomy. So most of the region has been segmented automatically, but some of them has to be segmented manually in mouse lemurs. So here I wanted to reproduce what I've been done here, but with a statistical comparison. So as I said, we created a method that we call statistical comparison of the relative correlation difference between functional and anatomical region, which sounds complicated, but basically it's the same thing as asking, when compared with medial prefrontal cortex, is the dorso-lateral prefrontal cortex more or less connected to the PCC? So what I've done is that I extracted in all my species the correlation between the PCC and dorsolateral prefrontal cortex in each individual, done the same thing between PCC and medial prefrontal cortex, and then perform a statistical test to see which one is greater than the other one. So when I've done that on human macaques and mouse lemur, for example, for the connection between the temporal and the dorsolateral prefrontal cortex and the medial prefrontal cortex, I obtained that the medial prefrontal cortex seems to always be more connected than the dorsolateral prefrontal cortex to the temporal area. However, if I compare human macaques, mouse lemur now for the correlation between PCC, dorsolateral prefrontal cortex, and medial prefrontal cortex, I found that the medial prefrontal cortex is strongly connected to the medial prefrontal cortex, but that was not the case in macaques and mouse lemur. So then, since the functional atlas might have some ROE problems since it doesn't follow the anatomical boundaries, I also repeated this test on the 2 atlases that have similar region in macaque and human. and obtained that the area 8AD is actually not less connected than the medial prefrontal cortex, but as connected in human. And if I, for example, repeat this test with the posterior anatomical area that is usually integrated into the default mode network, I obtain exactly the same results. And finally, in macaques, I obtain the opposite results, meaning that the 9M is always less correlated than the 8AD. So in conclusion, here we saw that it seemed that the PCC and the medial prefrontal cortex were always, the connection were always weaker in mouse, lemur, and macaques, but not in humans. We might have found a new network that involves the medial prefrontal cortex, and that seems to be similar to non-Hominid primate, but not to human. And finally, this area 8AD, that seems very interesting. Is it an ancestral characteristic of the primate frontal parietal network? So if this is the case, and we have two different networks, the hypothesis will be, do we have a reorganization or a fusion of the frontotemporal network or the frontoparietal network that could be a singularity of the human default mode network and potentially explain its specificity. So thank you for your attention.
Thank you very much. That was absolutely fascinating. And let me see if Shekoofeh has given me any questions. No, because we have a shy audience, and so we don't have questions. But of course, I have lots. So I'm going to ask you, Clément, a couple of questions. One big picture question, and that relates to you've got quite a difference in level between the human subjects and then macaque and mouse lemurs. And I want to get you to speculate about what you think might happen if you were to do the same thing for a great ape brain. What would you expect? Would they be more like the human one with the connection between the PCC and the medial prefrontal cortex? Or would you expect it to be more like the macaque?
That's a very interesting question. I mean, the day I would have this data set, I will be really happy to do the analysis. It appeared that only one paper has been published on the chimpanzee default mode network. And it seems that the medial prefrontal cortex is involved in the default mode network like of this primate. So that's why I chose the title as non-Hominid, because I don't want it to say non-primate, since there is this study that say that it might be not a human specificity, but a non-Hominid specificity.
Yeah, I wondered about that title because I thought there was a backstory behind it.
Exactly.
Yeah, great. Thank you for that. And yeah, if we find that paper later, maybe you can share that paper in the chat when the next presenters, because I'm sure other people might be interested in seeing that. And that goes too for the audience. If you know of any interesting papers that might relate to some of these studies, feel free to share those web links in the chat, because that's what makes these sessions online much, much more interesting for everybody. Now, I have a second question, which is a more nuts and bolts question for you, Clément, and that relates to these brains of these little primates, the lemur, the mouse lemurs, and the macaques, of course, are much, much smaller than the human brain. And so I'm thinking about the kinds of changes in blood flow, in the fluctuations, and the kinds of frequencies that these fluctuations occur on. And are they vastly different relative to, say, what goes on in the human brain? Because I know people in the human brain with resting state fluctuations have looked at wavelets type analyses and looked at the frequencies of that the various bold signals. What kind of frequency changes did you see in these very small primates?
I mean, I didn't actually really look at the comparison of the frequency, but yeah, since all the frequency were bandpass between 0.1 and 0.001. It's actually something, yeah, that could be interesting to see if there is some changes, because I know, for example, that studying the blood flow in mouse lemur is actually very difficult because it has a specific heartbeat. Or I'm not an expert on that, but I know that we tried in my previous lab and it was, yeah, almost impossible. So, yeah, it might be interesting to study that more in depth. Yeah.
Well, congratulations on getting a mouse lemur fMRI signal, given those difficulties. That's a tour de force, and that is just amazing. Beautiful data. Thank you so much for sharing that with us today. And so we will move.